Morning everyone. This is Gian's Academy, Ramabram, Chennai. We are giving online course on life science and all the other subjects also. In this video, we are going to see Unit 2 Cellular Organization in Life Science. Inside the cell, the membranes of the endoplasmic reticulum, Golgi apparatus, mitochondria, and other membrane bounded organelles in eukaryotic cells maintain the characteristics differences between the contents of each organelle and the cytosol. You have to know the eukaryotic cell and prokaryotic cell. See, what you are seeing here is the eukaryotic. There is a distinct nucleus in the cell, and in prokaryotic you will find only the cell membrane without a distinct nucleus. Okay, the genetic material will be placed in the cytoplasm. Actually, a cytoplasm is the plasm or the fluid which is between nucleus as well as the cell membrane. This area is called as cytoplasm, and inside the nucleus it is filled with the plasm called as nucleoplasm. Okay, if there is a distinct nucleus, then it is called as eukaryotic cell, and if there is no distinct nucleus, then this cell is called as prokaryotic. So this fluid is called as protoplasm. Okay, the nucleoplasm and the cytoplasm mixed together. So now this is called as protoplasm. Okay, this is prokaryotic and this is eukaryotic cell. Ion gradients across membrane established by the activities of specialized membrane proteins can be used to synthesize ATP to drive the transmembrane movement of selected solutes, or in nerve and muscle cells to produce and transmit electrical signals. See, I hope you understand. Look here, the ion gradients across the membrane in the sense the cell membrane. See, for example, if this is the cell and this is the membrane of the cell, if you see the cell membrane, the structure we will see later, this cell membrane consists of proteins as well as lipids. Okay, so the ion pass from the base area to the upper area, and from the outer side to inside and inner side to outer side. Okay, so that is what given here. Ion gradients across membrane, they cross the membrane and go outside, as well as from the materials from outside which crosses the membrane and come inside. How it is taking place we are going to see in this class. So it can be used to synthesize the ATP to derive the transmembrane movement. So the movement is called as transmembrane movement of selected solutes that happens in the nerve cells as well as the muscle cells. Neurons, nerve cell in the sense it is neuron, and muscle cells, you all know that. So there what is happening in the sense, the electrical signals will be given out, that will be received by the next cell. See, for example, this is another neuron in the sense this is releasing a signal, electrical signal, that will be received by the next neuron and it is passing to the another cell. So here what is happening in the sense, there is a through the transmembrane, the electrical signals are transmitting from one cell to another cell. Okay, for that the membrane is very important. The cell membrane is very important. Across the cell membrane, these signals are moving from one cell to another cell. Okay, and in all the cells, plasma membrane also contains the proteins that act as sensors. Okay, that acts as sensors and they send the electrical signal allowing the cell to change its behavior in response to environmental cues. These proteins are called as sensors or receptors. They transfer the information rather than ions or molecules across the membrane. Okay, they transfer the information from one cell to another cell, okay, through the plasma membrane. See what you are seeing here, the structure of the membrane. This is electronic microscopic structure. Okay, this is a dynamic fluid structure, and most of their molecules are to move about in the plane of the membrane. That is from, for example, the blood vessel is carrying so many nutrients. So those nutrients are coming into, this is inside the cell. If you think, this is the inner part of the cell. Okay, this is the membrane, outer membrane, and this is the blood vessel. If you think, the blood vessel carries all the nutrients as well as the oxygen, okay, oxygen and other nutrients that will come through this membrane. And now here the membrane allows these molecules to enter inside, as well as the carbon dioxide which is produced outside, which is produced by the cell, as well as the waste materials, that is nitrogenous waste, will be produced after the respiration, as well as the biological activities which is taking place inside the cell will be moved from inside to outside, and the nutrients which is coming from, nutrients as well as oxygen from the blood vessel, which comes through the membrane only. So here what it is, this membrane is made up of protein and lipids. Okay, so this is called as dynamic fluid structure. The cell membrane is a dynamic fluid structure, and most of the molecules are able to move about in the plane of the membrane. The lipid molecules are arranged as a continuous double layer. You can see here, this is one layer of molecules and this is another layer of lipid molecules, and the thickness is about 5 nm. The thickness is 5 nm. And this membrane structure, and it is called as fluid mosaic model. It is proposed by S.J. Singer. In the plasma membrane, see this is the plasma membrane. Some proteins serve as structural links that connect the membrane to the cytoskeleton. Cytoskeleton means a minute tubules which present in the cytoplasm. See, it's a net-like structure. Cytoskeleton means it gives support to the cell. Okay, just like our bones, it gives support to the cell. That is called as cytoskeleton. Okay, so some protein will attach with the cytoskeleton, and some other with the extracellular matrix with the adjacent cell. Okay, while others serve as receptors which detect and transduce the chemical signals in the cell's environment. They receive in the sense, the receptors, they receive the signals from the adjacent cell. A phospholipid bilayer with a hydrophobic, that is non-polar, okay, hydrophobic, and these are hydrophobic part of the membrane. Okay, you can see two layers, this one and as well as this one. Okay, this is outer layer and this is inner layer, this double membrane, plasma membrane. Okay, in that the outer membrane, outer side of the membrane, if you see, this part is hydrophobic and this part is hydrophilic. Okay, so this area is called as non-polar fatty acid chains, okay, which is directed inward, and the polar hydrophilic heads lining on the surface. Okay, this is hydrophilic and this is hydrophobic, right? And proteins, glycoproteins and lipoproteins associated with the inner and outer surface of the membrane. What is glycoprotein? In the sense, it is carbohydrate plus protein combined together, it is called as glycoprotein. And lipoprotein, in the sense, lipid plus protein, is called as lipoprotein. Okay, so all these are embedded in the cell membrane and make a cell membrane very effective. They do various functions. Okay, if you see this, this protein is embedded inside the membrane, and some part of the protein is extruded outside of the cell and extruded inside the cell, and some part embedded inside the cell membrane. Okay, the polar portion of the proteins associated with the polar surface and may protrude from it, while portions of the protein may embedded in the bilipid layer. This is what called as bilipid layer. Okay, and the non-polar portion, that is hydrophobic, this is called as hydrophobic. Okay, it is called as non-polar region. Okay, and proteins having both polar and non-polar residues are termed amphipathic. Okay, they have polar and non-polar polar and non-polar areas in the same protein, which is called as amphipathic, keep in mind. Sima, lipids of the membrane are fluid at physical temperature. It is fluid, the membranes, lipids of the membrane. Okay, you have to keep in mind, lipids of the membrane are fluid at physical temperature. Thus, they may be fluid movement of the lipid molecules and lateral movement, that is diffusion of the proteins that are embedded in the membrane. Okay, this is the singer, he is telling the proteins icebergs in a sea of lipid. Okay, these are the phospholipids, and the proteins are embedded here and there, so it is looking like an iceberg. So it is called as protein icebergs in the sea of lipid. It is by Singer. Okay, this forms of heterogeneous mosaic of proteins which may be continuously rearranged but often with specificity. So this is called as mosaic. Okay, it is giving an appearance of mosaic. So a singer, he quoted that this protein icebergs in a sea of lipid. See here, the exchange from one monolayer of the bi-leaflet. This is one leaflet and another one leaflet. So the exchange is flip-flop. Okay, this is flip-flop in the sense, from here the material is carried to that side, and from that side it is carried to this type. So from here to there, there to here, so that moment is called as flip moment. Okay, it is flip-flop moment, and there is an enzyme which is required for this moment. It is called as flippase. Okay, so the process is facilitated by a specific proteins, that is the enzymes, which is called as flippase. This is also one of the important question, so make a note. The membrane proteins are either extrinsic or intrinsic. Extrinsic membrane protein are entirely outside of the membrane but are bounded by weak molecular attraction, that is ionic, hydrogen and van der Waals bonds. Okay, so this membrane is called as extrinsic membrane protein. Extrinsic membrane and the protein which is involved here are called as extrinsic membrane proteins. Okay, and what are intrinsic membrane protein? In the sense, they are embedded in the membrane. They are embedded in the membrane, so they are called as intrinsic membrane. And transmembrane protein, in the sense, many of them extended from outside the membrane to the other are referred as transmembrane protein. These are transmembrane protein. Okay, these are extended, so these are called as transmembrane protein. Okay, so there are three membrane proteins: extrinsic membrane, intrinsic membrane and transmembrane. Okay, See, there is a question, how can ion be transported across membranes that are effectively impermeable to them? Okay, impermeable means this plasma membrane is selectively permeable. It allows only a certain important nutrient to come inside, as well as certain important material to go outside, not all. So they are called as semi-permeable or impermeable. Okay, effectively or selectively. And the cell contains proteins that are embedded in the lipid bilayer of that plasma membrane and extend from one side of the membrane through to the other. Okay, you can see here it is extended, and here you can see this also, it is extended, this is cut open. Okay, so it is having a path at the middle. Okay, so this is also extended, and some of the part are embedded in the membrane, cell membrane. Okay, such transmembrane proteins can function to effect ion transport. Okay, through this the ion transport takes place in several ways. See, there is a question, how can they cope with the energetically highly unfavorable situation in which an ion must pass through the hydrophobic inner layers of the plasma membrane? Okay, so this is a question. It's a very good question. We examine the detailed structure of many transmembrane proteins. See, what you are seeing here is a transmembrane protein. Okay, and they often have three different domains. Okay, see, this is one domain, this is another domain, and this is the third domain. So in this, two are hydrophilic. You can see this is hydrophilic, and that is also hydrophilic, and this is hydrophobic. Okay, the hydrophilic domain consists of hydrophilic amino acids at the end terminus is poking out the extracellular. See, what you are seeing here is end terminus is poking out in the extracellular medium. A hydrophobic domain in the middle of the amino acid chain, often only 20 to 30 amino acids long, is threaded through the plasma membrane. Okay, it is threaded through the plasma membrane, and a hydrophilic domain at the C terminus protruded into the cytoplasm. See, this is C terminus, and this is what we call N terminus. Okay, N terminus. This is N terminus, and this is C terminus. Okay, the C terminus is protruded into the cytoplasm. N terminus is outside, that is extracellular area it is, or extracellular medium it is protruding. Okay, and this middle piece which consists of 20 to 30 amino acids. Okay, 20 to, this middle piece is 20 to 30 amino acids long. Okay, 20 to 30 amino acids long is thread through the plasma membrane. It thread through the plasma membrane. This is the plasma membrane area. It threads through the plasma membrane, and a hydrophilic domain at the C terminus is here. Okay. Now the transmembrane domain, because it is made up of amino acids, because it is made up of the amino acids having the hydrophobic side chains. These are the hydrophobic. Okay, hydrophobic side chains exist comfortably in the hydrophobic inner layer of the plasma membrane, because these transmembrane domains anchor many proteins in the lipid bilayer. Okay, this is the lipid bilayer. So this anchor many proteins in the lipid bilayer. Okay, these proteins are not free floating, cannot be isolated and purified biochemically without first dissolving away the lipid bilayer with a detergent. Okay, so this cannot be isolated. Transmembrane proteins are glycoprotein. I told you already, the glycoproteins are carbohydrate and protein combined together called glycoprotein, which are covalently attached to their hydrophilic domain. See, this is hydrophobic domain, and these two, this is one hydrophilic and this is another hydrophilic domain. Okay, so it has three domain. One is hydrophobic and two hydrophilic domain. Okay, now a typical mammalian cell may have several hundred distinct type of glycoprotein shedding its plasma membrane. Okay, and if you see this, hydrophilic domain having hydrophilic amino acids. Okay, alternate with the hydrophobic domain. These two are hydrophilic, which possess or which contains hydrophilic amino acids. Okay, and this is hydrophobic. Now the protein chain as a whole embedded in the plasma membrane actually weaves back, okay, weaves back and forth between the opposite sides of the plasma membrane. Such proteins have the configuration of snakes. Okay, you can see here it is back and forth. It is just like a back and forth. It just, okay, it makes like a snake. Its appearance is like a snake, so it is called as the configuration of snake, and hence the term serpentine membrane protein. So this protein is called as serpentine membrane protein because of its appearance. Okay, a commonly used type of structure seen in many hundreds of serpentine transmembrane protein involves 7 hydrophobic domain inserted in the plasma membrane. Here you can find the 7 hydrophobic domain, okay, which is inserted in the plasma membrane, separated by hydrophilic region. They are looped out, that is this is external part of the cell, and this is internal, that is cytoplasm. It is protruded into the cytoplasm. This is extracellular or outside the cell wall. Okay, sorry, cell membrane. Outside the cell membrane it protruded. Okay, so this is one hydrophilic, this is another hydrophilic. So 2 hydrophilic and 1 hydrophobic. This protein is called as transmembrane protein. It is made up of glycoprotein. Okay, and the name it takes serpentine membrane. Okay, serpentine membrane protein because of its shape. Okay, this is also very important. Let us recall this. Plasma membrane also contains protein that acts as sensors of external signals. These protein sensors or receptors transfer information rather than ions or molecules across the membrane. The lipid molecules are arranged as a continuous double layer about 5 nm thickness. Membrane ultrastructure and fluid mosaic model is proposed by S.J. Singer. Protein icebergs in a sea lipid. This quotation is given by Singer. Some proteins serve as structural links that connect the membrane to the cytoskeleton. Proteins having both polar and nonpolar areas are termed amphipathic. Exchange from one monolayer of the bileaflet to the other monolayer. This process is facilitated by specific proteins, that is enzymes called flipases. Membrane proteins are either intrinsic or intrinsic. A hydrophilic domain consists of hydrophilic amino acids. At the end terminus is poking out in the extracellular medium. Hydrophobic domain in the middle of the amino acid chain, which contains 20 to 30 amino acids long, is threaded through the plasma membrane, and a hydrophilic domain at the C terminus protrudes in the cytoplasm. A typical mammalian cell may have several hundred distinct types of glycoprotein studding its plasma membrane. Proteins are the configuration of snakes, and hence term them serpentine membrane proteins.